It's my pleasure now to introduce David Suter. David completed his MD PhD at the University of Geneva and continued to do postdoctoral research in Geneva with Uli Schibler, where he developed a bioluminescence imaging approach to study transcriptional bursting, and then moved to Harvard for a second postdoc with Sunyi to continue working on transcription factor dynamics and single molecule imaging in mammalian cells, and established his laboratory at the Institute of Bioengineering in the School of Life Sciences at EPFL in Lausanne, Switzerland, just over 10 years ago now. And his laboratory has been doing some really cool work using a variety of different tools and genomic approaches to continue studying transcriptional dynamics and gene regulation. And I'm excited to hear his talk today, where he will be telling us about H2A.Z and SRCAP. So David, please take it away.
Okay, thank you very much, Jackson, for the kind introduction. And thank you for having me here. I'm super excited to be part of this series. Yes, so indeed, today I'm going to talk about a histone variant called H2A.Z and the chromatin remodeler that loads H2A.Z on chromatin, that is SRCAP. So, sorry, it's not moving. Oh, yeah, now. So I'd just like to give you a few introductory points about the H2A.Z histone variant. So H2A.Z is a variant of H2A that is highly conserved from yeast to mammals, but is quite different from H2A, suggesting that it has acquired a very different function from the canonical H2A. H2A.Z is absolutely required in mammals during early embryonic development. So if you don't have H2A.Z, the embryo doesn't implant. So you can find H2A.Z at inactive and also bivalent and poised promoters and enhancers. So these are regulatory regions that either produce transcripts or that are poised to be activated during early development, for example, in embryonic stem cells. So as I mentioned, H2A.Z is mainly deposited by the SRCAP complex that is also highly conserved from yeast to mammals. And finally, I'd like to mention that H2A.Z has been correlated with more unwrapped and also fragile nucleosomes, and also shown to be anti-correlated with DNA methylation, which has led to the suggestion that it may actually inhibit DNA methylation or favor DNA demethylation. So the role of H2A.Z in regulating transcription is still not totally clear. And one reason for that is that most studies that have been targeting H2A.Z have used approaches that are relatively slow, such as knockout or slow knockdown using shRNA. And of course, gene regulation occurs on much faster timescales of minutes to hours. So it's not totally clear whether these studies really allow to assess the direct effect of or implication of H2A.Z in transcriptional regulation. Another potential issue if you deplete cells from H2A.Z is that it's not totally clear what happens to H2A.Z containing nucleosomes. Are these replaced completely by canonical nucleosomes? Do we get some regions without nucleosomes? And also, is there any function of soluble H2A.Z? Another potential confounding factor when depleting H2A.Z is that it itself also impacts the recruitment of its own chromatin remodeler. So in yeast, if you knock out H2A.Z, what you actually see is that the yeast version of SRCAP that is called SWR1, let me just get the laser pointer. So if you deplete H2A.Z in these conditions, you actually see that the SWR1 is binding much less to the promoter regions where it's normally heavily occupying in yeast. And another more recent study has shown that when H2A.Z is incorporated in nucleosomes, it actually repels SRCAP as compared to canonical nucleosomes. So this tells us that each time you touch H2A.Z levels, you may also alter the binding landscape of its chromatin remodeler. And now whether SRCAP has functions in gene regulation that are unrelated to H2A/Z is not entirely clear. So now the question is, how can we study the specific and direct function of H2A/Z with respect to H2A on gene regulation and the function of SRCAP that is independent of its H2A/Z depositing activity? So to study this, we decided to use mouse embryonic stem cells as a model system. So mouse ES cells express SRCAP at high levels. They also have high levels of H2A/Z on the chromatin. As you probably all know, ES cells can either self-renew in culture, or they can also differentiate towards the different germ layers. And they also have a lot of these genes that are called bivalent or poised, and many of them are actually important for lineage specification as they encode transcription factors that regulate these developmental events. So to try to solve previous challenges with the study of H2A/Z, we decided to use combination of two strategies. So SRCAP its job is normally to swap H2B, H2A dimers for H2A/Z, H2B dimers. So we reasoned that if we could rapidly deplete SRCAP, we would shift the equilibrium of H2A/Z nucleosomes toward the canonical nucleosome, and that this could allow to now really study the function of H2A/Z versus H2A as we swap both types of nucleosome in the same positions. And then, so this approach would, if it works, it will both swap the nucleosome types, but also deplete SRCAP. So now to distinguish between the function of SRCAP and H2A/Z, the second approach we combine with this is to rescue SRCAP depletion with a catalytically dead mutant SRCAP that cannot deposit H2A/Z, and in which we made a 3-point mutation in the ATPase domain. So I'm first going to explain a bit the first approach and show you some results before showing you what we can actually learn from combining this with a catalytically dead SRCAP. So to deplete SRCAP rapidly in mouse ES cells, we use an auxin-inducible degradation system. So we fused both endogenous copies of SRCAP with an auxin-inducible degron and a fluorescent protein to seed. And we also co-express the ubiquitin ligase that allows to degrade proteins that carry this degron in the presence of auxin or IAA. So you can see that within about 2 hours, we get almost full degradation of SRCAP with this system. And from now on, I'm going to refer to this cell line as the SIAD cell line. So now the next question was, what happens to H2A/Z and H2A if we deplete SRCAP? So we expected to see this swap from H2A/Z to H2A nucleosomes, but we didn't really know beforehand what would be the timescales of this swapping. So we took our cell line that we treated with auxin for different time durations, and we performed both H2A/Z and H2A ChIP-seq. And what we found is that H2A/Z is rapidly depleted from chromatin. And if we look at the H2A content at the same positions where we have, centered on the H2A/Z peaks from the control, what we see is that these regions are initially depleting in H2A because they have H2A/Z there, and then they get progressively filled up with H2A. So this shows that if you rapidly degrade SRCAP, you actually swap H2A/Z for H2A within a few hours. And so if you look at gene regulatory elements I mentioned before, active promoters, active enhancers, bivalent and post promoters and enhancers, you see that this decrease in H2A/Z is also on globally in the same time scale that I showed you on the whole genome, and even a bit faster at active regulatory elements. So now with the system at hand, we wanted to understand what are the transcriptional changes that happen when we deplete SRCAP rapidly. So for this, we performed nuclear RNA-seq, and we analyzed intronic reads to get as close as possible to transcription. And here you see a time-lapse volcano plot that shows a progressive dysregulation of transcription. So as you can see, we have both downregulated genes, and these were essentially housekeeping genes. And we also had a more or less equivalent number of upregulated genes. And these were very highly enriched in lineage-specific regulators. So we had a lot of transcription factors in there, and also other proteins important for lineage specification, such as DNA methyltransferase. So now the next big question is, okay, how does this work? How does SRCAP loss lead to transcriptional changes? So in the course of this work, we've been looking at a lot of different parameters, and but in the interest of time, I will not go in detail through all of these parameters. But just to very briefly summarize what we saw at the level of the first 4 parameters I'm listing here. So we saw changes in both activatory and inhibitory histone marks and promoters. We saw changes in chromatin accelerated enhancers, but these were not really related to the change in transcription as far as we could tell. Surprisingly, we didn't see major changes in DNA methylation, suggesting that at least in the very short term, simply removing H2A.Z from chromatin does not elicit a rapid change in DNA methylation. So if there is any causal link between these two, it's probably occurring on longer time scale. We also looked at nucleosome positioning and fragility, and here we also didn't see major changes. Even though H2A.Z was associated with fragile nucleosomes, we actually even saw a slight increase in fragility when H2A.Z was replaced with H2A, which was also surprising to us. But so, yeah, I will not talk too much about this in detail, so you can find more details in the preprint that we published this year. But now I would like to spend the rest of the talk discussing changes in transcription factor binding that we observed. So the first way we looked at transcription factor binding in a very broad manner was to take advantage of ATAC-seq. So as you probably all know, ATAC-seq allows you to map accessible regions of the genome by taking advantage of a transposome that is loaded with sequencing adapters that lends into active open regions of the genome. And then you get these peaks on the genome tracks that tell you where the genome is accessible. But now if you zoom into these peaks, you can also see little dips that are caused by the binding of transcription factors that leave these footprints. And in these footprints, you can look at the transcription factor motifs that are enriched. So this tells you something about transcription factor binding in a genome-wide scale and agnostic of the transcription factor you're interested in. So now we could compare this footprinting between the control sample and the samples where we deplete SRCAP. And so what we found is that after 4 hours of auxin treatment, we had a bit over 100 transcription factor footprints that were decreased in their intensity. And we also had actually way more transcription factor footprints that were actually increased in the absence of SRCAP. And also interestingly, if we look at the type of transcription factor that binds to this motif, we found a lot of pioneer transcription factors. So transcription factors that have been shown to be able to bind to nucleosomes. So then to confirm these results, we collaborated with Enes Ugur, postdoc who was at the time PhD student in the Leonhardt lab in Munich. And so they have developed a method that allows to analyze the proteome that is bound to chromatin, so the so-called chromatome. So the basic idea is to cross-link proteins to chromatin, then to sonicate this, to then extract the proteins, digest these proteins, and finally do mass spec on these proteins. So this allows you to have a broad view on the proteins that are bound to chromatin. So of course, you don't see absolutely all proteins. You're a bit biased towards the most highly enriched proteins. And so transcription factors are typically expressed at moderate to low levels, so you only see some of them. But nevertheless, if we look at the proteins that are enriched or depleted by in our cell line when we treat cells with auxin, we saw that there were way more transcription factors that were enriched on the chromatome than depleted. And if you look at who are these transcription factors, these are the same that either increase or decrease the footprint. And then we further validated these results by chip sequencing on a few candidates. So for example, YY1 was a transcription factors that had decreased footprint and that was depleted on the chromatome. And what we saw is that when you treat cells with auxin at most genomic loci where YY1 binds, you actually have a decrease in the intensity of the ChIP-seq peaks. And conversely, for transcription factors that were enriched in the footprinting or enriched in the chromatome, we saw that you had more regions with increased binding of these transcription factors. So now with this data at hand, we're pretty convinced that upon SRCAP loss, we have major changes in transcription factor binding. But what we don't know yet is whether these changes in transcription factor binding and also those changes in transcription activity that I showed you before are caused by the loss of H2A.Z on chromatin, the loss of SRCAP, or both. So to try to solve this issue, we then took advantage of this catalytically dead SRCAP mutant that we generated. And so the way we implemented this mutant is to basically start with our SIAD cell line that allows degradation of SRCAP. And then in this cell line, we either express the wild-type version of SRCAP tagged with mCherry, or in another subcell line, the catalytically dead version of SRCAP also fused to mCherry. So now these cell lines can be treated with or without auxin, and we can then fax the cells for the same mCherry level to make sure that we are really comparing cells that have the same levels of SRCAP. But what we expected to see is that if we express the catalytically dead SRCAP, we would actually not restore H2A.Z levels if we treat cells with IAA, while the wild-type SRCAP should rescue these levels. And this is what we see here. So we see here the control SRCAP that is now, the cell line that expresses SRCAP wild-type, either with or without IAA. And you can see that SRCAP rescues H2A.Z peaks, while the catalytically dead does not rescue this H2A.Z on chromatin. So now we can compare head-to-head the rescue with the catalytically dead treated with auxin versus the wild-type SRCAP treated with auxin. So now we're in a situation where in both cell lines we have the same SRCAP levels, but in one case we rescue H2A.Z levels and the other case we don't. So this allows us to see changes that are caused by H2A.Z and not SRCAP. And so here what we're looking at is, again, an analysis of intronic reads in nuclear RNA-seq. And what you now see as compared to the first plot I showed you is that instead of having both down and upregulated genes, we have mostly upregulated genes in this case. And these genes are by and large the same that we saw when we degraded SRCAP in the original experiments. So this basically tells us that H2A.Z is a repressor of the expression of these genes. So when you lose H2A.Z, you get this upregulation. And by combining this data all together, also with the cell lines without auxin, we could infer the number of genes that are upregulated or downregulated by either H2A.Z or SRCAP. And what you see is that indeed H2A.Z inhibits transcription of most genes, while SRCAP is actually stimulating the transcription of the genes. So all the genes that you saw downregulated in the first experiment of SRCAP loss were downregulated because SRCAP was lost, not because H2A.Z was lost. And so this impact of H2A.Z on transcription is well in line with this work from Laurie Boyes' lab, where they showed that if you deplete H2A.Z from cells, you actually accelerate RNA-2 post-release, so you increase the transcriptional output of a number of genes. Okay, so what about changes in transcription factor binding? So the way I'm showing this analysis here is the following. So here, each dot you see is a transcription factor footprint, and we are correlating actually two different comparisons of these footprints. So on the x-axis, you have the original cell line that is treated with auxin, so that is depleted of SRCAP and is also depleted of H2A.Z. And we compare this to the rescue with the SRCAP wild type. So we restore SRCAP levels, but also H2A.Z levels. Now on the y-axis, you have again the Sayat cell line treated with auxin, but now we compare this to the SRCAP that is catalytically dead, also treated with auxin. So in this vertical comparison, you restore SRCAP levels, but you don't restore H2A.Z levels. So now this means that if H2A.Z was regulating, was the main regulator of this change in transcription factor binding, you should see most dots aligned on this horizontal line, because this is where H2A.Z levels change, only on the horizontal axis. But in fact, as you can see, most dots actually align on this diagonal, and this corresponds to the SRCAP regulated footprints, because in both axes, SRCAP is actually changing. Okay, so now the next question is, okay, how does SRCAP regulate transcription factor binding? And as I mentioned before, when you remove SRCAP, you have mostly upregulated binding of transcription factors, which means that SRCAP is mainly inhibiting transcription factor binding. So to try to answer this question, we looked at the crystal structure of SRCAP together with the nucleosome. And so you can see here SRCAP in purple that is embedded here in the whole SRCAP complex that has 9 other proteins. And you can see that this is an extremely bulky complex that is literally covering a large part of the nucleosome. And as far as I know, this is pretty unique among chromatin remodelers. And if you try to compute the accessibility of DNA within this structure to transcription factors, you can see that actually a large fraction of DNA is not accessible to transcription factors. Another interesting aspect of SRCAP biology is that it was shown to bind to the nucleosome for a very long time, for several minutes, at least in vitro. And in our ESLs, SRCAP is extremely highly present on chromatin. It's in the top 5% of most abundant proteins on the chromatome. So we thought that, okay, maybe SRCAP is, since it's very abundant, it's actually mainly on nucleosomes that are in relatively open regions of the genome. So maybe it could inhibit transcription factor binding by simply sterically hindering their access to DNA. So if this is true, we should find SRCAP in cells also mainly on nucleosomes. And so we perform SRCAP cut and run that we align on our MNA-seq data. And you can see that SRCAP is well centered on nucleosomes. And if we look at the transcription factor motif that where the footprints increased upon auxin depletion, we find that SRCAP is also well centered on this motif. Okay. So another thing you would expect if this is really steric hindrance is that this would act extremely fast. So we also checked whether SRCAP loss for only 2 hours would lead to an increase in transcription factor binding. And in terms of footprinting, we see virtually the same picture than 4 hours, okay, suggesting that indeed this is extremely fast. And here is an example of ChIP-seq for NANOG after 2 hours of SRCAP depletion that goes up immediately. So then the final thing I would like to discuss is the fact that transcription factor occupancy is actually, I mean, all what we showed so far is changes in occupancy. But changes in occupancy, they can be driven by different things. So you have transcription factors that go on and off DNA or nucleosomal DNA. And an increase in occupancy can be mediated by 2 mechanisms that are not mutually exclusive. You could have an increased residence time of transcription factors. So the off rate from DNA would be slowed down, so they would stay longer. Or you could have an increased binding frequency. okay, transcription factor coming more often, finding more often the target. And so we reasoned that if it's really steric hindrance, okay, either the transcription factor can bind to the nucleosome, or when SRCAP is there, it cannot bind at all. So we're expecting that steric hindrance should decrease binding frequency, but not residence time. Well, in contrast, if you would have a change in the, for example, chemical composition of histones on the nucleosomes, you would expect a change in affinity of binding for the transcription factor and a change in residence time. So to discriminate between these scenarios, we collaborated with the team of Christoph Gebhardt in Ulm. And so what we did is to generate a cell line that expresses nanog tagged with a halo tag at low levels in mouse CS cells. And then using a special elimination scheme that I will not explain in detail now, you can actually track single molecules of nanog in the nucleus. So what you see in these movies are nanog molecules moving around. And when you see these bright dots that stay a bit longer, these are nanog molecules that are bound to DNA. So with this kind of data, you can now measure the bound fraction of nanog to the genome. And that's in a way equivalent to ChIP-seq data. But you can also now differentiate changes in binding frequency from changes in residence time. And so what we found is that when you deplete SRCAP, you increase the bound fraction in a quantitatively very similar manner than what we saw in ChIP-seq. And this is rescued by the expression of CD SRCAP. If you look at the binding frequency, so this is the parameter that really increases when you deplete SRCAP, fourfold increase in binding frequency. But when you rescue this with the catalytically dead SRCAP, this goes back to the same level as in the control. And the residence time is actually even going a little bit the opposite way as compared when you deplete SRCAP. And this is probably because when you remove SRCAP, nanog starts to bind at many more places where it has less cofactors to bind, so where it may on average bind for less time. Okay, so this brings me to the conclusions of my talk today. So I've shown you that H2A.Z is essentially restricting transcription of lineage-specific genes. We found that SRCAP has two catalytic independent functions. It stimulates expression of housekeeping genes, so we still don't know how this works. And it also sterically hinders pioneer transcription factor binding. And what we find really interesting with this couple is that, in fact, the transcription factors that are inhibited in the binding by SRCAP are a lot of lineage-specific TF. And many of these are expressed at low levels in ES cells. And many of these are the same where H2A.Z plays a role in restricting their expression. So it seems that these couples have evolved to restrict both the binding of these transcription factors and also the expression of these lineage-specific regulators. Okay, so I would like to thank the people who were involved in this work. So first, Armel Tollner, very bright postdoc in the lab who did the vast majority of this work, who worked with Cédric Delu, lab manager, and already mentioned our collaborators in Munich, Enes Ugur and Heinrich Leonhardt, and here are our collaborators in Ulm University. And that's the preprint. If you're interested, there is way more in the preprint that I could tell you today. And thank you very much for your attention.
All right, awesome, David. Thank you so much. We've already got a few questions in the Q&A, so let me pull this up. Let's see. So we've got a couple questions about some of the other chromatin remodelers that may be playing a role in H2A.Z deposition. Deepthi asks if you can comment on the contribution of EB400, TIP60, I guess, in...
Right, yeah. So we didn't look directly at that, but as you can see here, so H2A.Z levels, they don't go to zero, right, when we remove SRCAP. So this is basically, this is centered on all H2A.Z peaks. So it goes down to significantly, but not to zero. So these are probably places where TIP60, P400 also play a role, but we didn't really investigate that further.
Sure. And then Jacques Hotz says, very interesting data on SRCAP, striking. For the non-correlation with H2AZ in yeast, it's known that in the absence of H2AZ, SWR1 starts doing weird things, explaining why the ZKO has a different phenotype than SWR1 KO. Could that be the mechanism here? Like, is it stuck on nucleosomes, or is this directly through blocking the two transcription factor binding?
So if I understood correctly, you're referring to the catalytically dead version that might be stuck on nucleosomes, right? Right. So the thing is that if we look at the footprinting analysis, basically the wild type SRCAP and catalytically dead, they do exactly the same thing. I mean, there's little small differences because H2AZ plays a little bit of a role in refining TF binding, but it's actually quite minor, right? So we don't think they behave much differently in terms of binding to the nucleosome because we have, I mean, very similar results with the wild type and the catalytically dead.
Then let's see, Olga Rospopov asks, is there a skew towards a specific lineage in the transcription factors or the other genes that you see being dysregulated when SRCAP is inhibited?
Yeah, very interesting point. So actually, we see transcription factor transcripts going up a bit all over the place. There is a bit more towards the mesoderm, but we also see transcript for pluripotency transcription factors going up. So it seems that it's not super directed. And what people have seen is that if you remove H2AZ or SRCAP, cells seems to be lost. They are both not very good at maintaining pluripotency and at differentiating. So it's not really, it doesn't seem to be involved in a very specific differentiation path.
Okay. Jane Skok asks if H2AZ depletion affects CTCF binding.
Right. So, okay. So keep in mind that we didn't really deplete H2AZ here, both SRCAP and H2AZ. But yeah, I mean, this is still something we're investigating. We started to do ChIP-seq for CTCF, and it seems to increase a bit. But yeah, so take this with a pinch of salt. These are the preliminary results.
Yeah. Alan Underhill raises an interesting point that H2AZ has a role in stimulating H4K20 methylation catalysis by KMT5B. Yeah. With that marker, with any other histone marks like acetylation across H3 and H4, do you see any crosstalk between SRCAP, H2AZ, and the other marks?
Yeah, so we did not look at this mark specifically. What I can tell you is that H3K4-trimethyl is going down dramatically, but it's not explaining changes in transcription. H2AK119 ubiquitin is going up, also not correlated to transcriptional changes, and H3K27-trimethyl is going up and down. So yeah, it doesn't really answer the question about 2 marks, but these are the ones we looked at. And we couldn't really figure out, and they were not really correlated to changes in transcriptions.
Right. And so those are the two different polycomb marks that you see going up when you.
Yeah, only the UB is going up a lot, but the H3K27-trimethyl, it's less marked and it's both up and down, depending on the region you're looking at. Yeah.
I see. Yeah. So I was wondering that besides transcription factors in your chromatome analysis, do you see like an increase in acidic patch binders that potentially SRCAP was blocking, or do you see more chromatin remodelers coming into the area to interact with the nucleosomes?
I mean, not really in terms of remodelers. I mean, the only marked change we see is the other members of the SRCAP complex. They are basically gone also from chromatin, but we didn't really see major changes in other chromatin remodelers. So yeah, it's mainly transcription factors that we can see,
sure.
And then a bunch of other proteins that we have to figure out actually what they're doing.
Sheila Teves asks if there are differences in chromatin binding between the wild type and the catalytic dead SRCAP.
Right. So we didn't look into that specifically so far, and the reason for this is practical. So for the wild type SRCAP, we had the wild pet tag on the endogenous, and we had a good antibody for cotton tag and cotton run. So this we didn't have for the catalytically dead. But again, since we saw very similar changes in footprinting, we assumed that it's probably binding to more or less the same places. But you're totally right. This is something that we should find a way to do, and we couldn't do that yet.
Do you know, are there any pharmacological or are there any inhibitors you can use to inhibit the catalytic activity of SRCAP, or have we not progressed to that point in chemical design yet?
Yeah, we've not investigated this. Yeah, that would be great to reach even higher temporal resolution indeed, right? And also
those of us that work with BRG1 are spoiled with our
right, yeah. And also separate with the function of non-catalytic function, yeah. I agree, but yeah, we didn't work on that yet.
Yeah, we need to get the chemical biology people to start focusing on more of these chromatin revelers, help us out, make our lives easier.
Right.
Well, David and Cassit, I'd like to thank you both again. Two wonderful talks, and they've mentioned David's new preprint is out there where you can learn more, and Cassit's work was just published. So please check them out, and please visit us again in 2 weeks when we have another 2 fascinating talks. Thanks again, David and Cassit, and everyone for attending. Have a great rest of your day.
Thanks. Cheers.
Thank you so much. Bye-bye.